Greetings. I'm Yvonne, Staff for Science for the Public, and I welcome you to Contemporary Science Issues and Innovations. For the past several years, due to the COVID pandemic, viruses have been a major topic and definitely a lot of misinformation and disinformation. Today, we get the real facts. Our guest is John Connor, Associate Professor of Microbiology at the Boston University School of Medicine and an Institute investigator at the National Emerging Infectious Diseases Laboratory, also known as NEEDLE. Dr. Connor's lab specializes in one of the most fascinating battles in nature, how viruses attack cells and how immune systems fight back. Dr. Connor, thank you for joining us again and a big welcome.
Thank you. It's great to see you again.
And to start with, a couple of things about viruses just in general. They're often confused with bacteria, but there is a difference. Would you tell us the difference, please? And then the issue of whether they're really alive or not.
Yeah, so the basic answer to what the difference is between viruses and bacteria, or viruses and fungi, or parasites, is that viruses are genetically more simple. They have less genetic material. They are different from the other infectious diseases that we encounter in that they are obligate parasites. So they do not have the ability to make copies of themselves outside of a host, whereas bacteria and fungi and parasites often do.
Okay, great.
The question of whether they're alive is a really healthy one. I come down on the side that they are not.
Okay. They're intelligent non-life, is that it?
They have become very good replicating machines.
They really are amazing.
They have the ability to very easily get into cells of hosts and then make copies of themselves and do that over and over and over again. But they can't do that without a host.
Yes, okay, I understand that then. Now, then the next thing is that it's a matter of confusion is RNA and DNA viruses. They don't have both, correct, as cells do. So, but RNA viruses seem to be very clever. Can you tell us the difference?
Yeah, so I would argue that all the viruses that are human pathogens end up being very clever.
Okay.
They do change what their strategy is and how they encode their genomes. There are viruses that encode their genomes as RNA. There are viruses that encode their genomes as DNA.
I see.
One of the remarkable things about viruses that encode their genomes as DNA is they're usually much larger than the viruses that encode their genomes as RNA, though that's not always true.
Okay.
It is two different strategies for keeping the genetic information of the infectious disease.
Okay. Now, do RNA viruses mutate faster, or is that what seems to be the case in some of these diseases, or is that not true?
RNA viruses do have the capacity to change rapidly. Mm-hmm. One of the facts is that they are shorter than DNA viruses. So if each virus makes a mistake once every time it makes a copy of itself, the RNA virus has an overall larger percentage change. So one of the things we've seen, and we've seen this with COVID, we've seen this with other RNA viral diseases, is they do change fairly rapidly. But that does not mean that the DNA viruses are incapable of changing rapidly. They can also change in response to threats.
I see. But we've just had such an abundance of these fast-moving, fast-mutating RNA viruses, so that's good to know. Now, you're a real expert on how they do their invading. Can you give us an idea of how they do this? They're really very clever.
Yeah, so viruses viruses package them up into they package themselves up into individual infectious units called virions. those virions have what is, in essence, a key on the outside that allows them to get into a cell, one of my cells, for instance. And once they use that key, they insert their genetic material. Their genetic material then drives a program where they now make copies of that genome. They make proteins that are virus specific. They also make proteins that get in the way of the cell responding, right? The cell is being taken over, but it's really not that comfortable with that idea. And there are internal cell programs to try to fight off viruses, but viruses have developed countermeasures for that.
Yes.
Once the virus has made copies of its genome, it's made copies of its protein, it then has an organized program to assemble up brand new virions that then leave the cell and start that cycle over again. And that can happen in an ever-expanding way that can cause real damage.
Yeah, that's just amazing that they are able to invade, pick out the right place to hit the cell, and then make off with the material that they need. These viruses, Dr. Connor, don't attack everything. It seems that in these cases where the viruses, like for coronavirus, in fact, I asked you a question when we started out with the COVID that a listener had asked, well, do we have to watch out for rats? For example, will they contaminate rats and then spill to humans and so on? And you said that's not going to be a problem, likely. But they do attack certain species and not others. What's the mechanism there, or the reason?
Yeah. Right. So the mechanism that explains that is that key that's on the outside of virions. In many cases, just like the keys we have, they don't work on everything. Viruses often have keys that will only get them into specific cells.
And
the other way that viruses can fail and cannot be successful is I mentioned that they have a way to try to get in the way of a cell's defenses. Sometimes those approaches are very specific for either humans or rats or mice, but don't work in a different host. And so there's multiple ways at which a virus can fail in terms of trying to infect another organism that it doesn't normally adapt itself to infecting.
I see. So they have to be a little selective in an odd way. They can't just hit everything that is out there. And then in another case here, we have this situation called spillover. And when we talk about COVID, there was the sense that that had been a spillover virus. Yeah. Could you explain what that is? What do we mean by that?
Yeah, so a spillover event is when a virus that is normally infecting and making copies of itself in one organism spills over, right? Goes into a host that it doesn't normally live in. A recent example is the MERS coronavirus, which appears to normally be passed in camels. However, under certain rare circumstances, the virus can be passed from camels to people. And so that's a spillover event. That's a virus that is normally circulating in one circumstance, now being introduced in a new circumstance.
I see. And in these cases where bats evidently, and that's something I'd like to pursue a little more, certain bats tend to carry a number of these particularly awful viruses, but they are known to be meat in certain areas of the world as well, certain animals. And so these viruses are then transmitted to humans in those cases with these meat markets. Is that true? And was that a situation in China or, well, in other parts of the world too?
Yeah, so the question of bats being carriers of diseases that we get, we've known that for years, right? Rabies is carried
Yeah.
by bats. We also know that a virus called Nipah virus is carried by bats, and good. They're very good examples of how you get those viruses passed from bats to humans. There are other more speculative suggestions, and you mentioned one of them, that there may be transmission through eating bats. I think that is not as well established.
But
But it's absolutely the case that bats do carry viruses that we can get. The
yes.
more we come into contact with them, the more possible it is that we will pick up a virus from a bat that we would not normally pick up if we didn't interact with them as often.
I see. And do these open, or well, I guess they're called wild meat markets, and they're common in many parts of the world, so that wild animals are killed and then brought to the market, or brought to the market and then killed, and they're in, more heavily populated areas, so that viruses can go from these creatures to humans or whatever. Is that in truth a common thing?
So
Markets.
the question of how common it is, I think, is not really known, but the overall idea that if you are bringing animals from the wild where you don't know what they've come into contact with, and you bring them into an environment where there are a lot of people, it does generally increase the chance that you will have a spillover event.
Okay.
You can imagine a similar thing if you went to a wild petting zoo.
Yeah, right, I suppose so. Or if we don't have like wild deer markets and things like that in this country, but that it would be a possibility to transmit these viruses if we did in a situation like that. There's one other thing here on where these things come from, and that is, a question has been raised about zombie viruses, which might emerge that have not been around for ages and ages because of the melting permafrost, for example. Is that a possibility?
Yeah, in a theoretical sense, the idea that there will be some very old viruses that are woken up as the permafrost thaws is almost certainly the case. Is that something that I lay awake at night worrying about? I really don't.
Right.
Right. The things that I think are going to wake up from the permafrost will be huge dangers to amoeba and other single-celled organisms. And maybe they should be frightened and afraid, but while it makes for a great movie plot,
Right.
I don't think that the permafrost is holding major animal-killing viruses
Right.
that we have a great deal to fear.
Great. That's a nice relief. Thank you. Because, as you say, it makes a good movie, but maybe not something that we
Right.
should be losing.
It would be a great movie.
I can see you writing out that script right now then. Okay. But we don't have to worry so much about that. The interesting thing there, in what I've seen, is that something could be dormant for, hundreds of years and then come to life like this. That would be a weirder thing, perhaps. But in any case, now, another issue for us that came out during COVID in particular was this thing about, just let the virus go, we'll develop natural immunity. And I think virus experts like yourself might have been a little nervous about that. I'm not sure. What can you tell us about that? What is meant by natural immunity, and does this really work?
So the idea of natural immunity is that if you have an infectious disease and it runs its way through a population, that whoever is left after the disease runs its way through the population will have some level of immunity. And that was happened through infection. That happened in a fairly natural manner. And the idea that that would happen is generally true. However, it does not take into consideration the fairly advanced nature of our society currently. One of the reasons that we have vaccines is it means that a lot more people survive diseases that would not have otherwise.
Yes.
A good example is measles,
Yes.
where the number of fatalities from measles in the United States is very, very low because been a fairly successful vaccine campaign.
Yes.
Because there has been a fairly successful vaccine campaign. In other countries where there is not a successful measles campaign, there is significant loss of life from measles. And so the reason that vaccines can be really good is, yes, you have to have a vaccine, but it can reduce overall suffering.
Right. And numbers, the fatalities, of course, did go down after people got vaccinated against COVID and so on. But we really are still fighting this battle against really disinformation about you don't really need these vaccinations at all. So I'm glad that you've put in your 2 cents on that, that we do need them and they are very helpful. I would like now to go to your research in general, and would you tell us, to start with, what you work on in particular? I know you've done an awful lot of work on Ebola, but other things too. What's the core of your research, sir?
So I think there's probably 2 things worth talking about at the moment. One of the things that we have been interested in for a long time is trying to develop new small molecules that could be developed as antivirals. And we have been focusing in the last few years on understanding compounds that will target pox viruses. Things like mpox and smallpox and other diseases caused by this family of viruses. And the work that we've been doing is really focusing on one class of molecule that we discovered and have found have really broad activity against all of the orthopox viruses that cause human disease. Wow. So at this point, we are identifying the most promising small molecules from that class, and we are developing them to see if they work in animal models of disease. The long-term goal of this is to add to the armament that we have that we could deploy in the event of more mpox transmission, in the event of spread of cowpox or other diseases that are caused by the pox virus family.
Oh, I see. You sort of specialize on the pox viruses and developing for those. Are there the other ones included too? I mean, does the bioengineering work as well for other varieties of viruses, families of viruses?
Yeah, so recently we have not been doing a lot of bioengineering of viruses. We have been doing a lot of work in tracking virus evolution. I think it's no surprise to you that viruses evolve. This became a very hot topic during the peak times of COVID transmission with the appearance of the Alpha, the Delta, and the Omicron variants, which were were SARS-CoV-2 or COVID, that has evolved over time. We were involved in helping understand how the virus was transmitting within this city, in particular, understanding how transmission happened in a university setting. And we were particularly interested in understanding not only when transmission happened, but when it didn't happen. Because that's one of the questions that people always have is, where is my risk? As you can imagine, in the fall of 2021, as students began to come back to class, there was an open question of, if I go to class, am I at risk of getting disease just from going to class? And that was a question we were interested in and were able to really try to directly understand. And what we found was that, at least at Boston University, where there was a vaccine mandate, where there was a requirement to wear a mask in class, but no requirement for social distancing, we didn't see any evidence of classroom transmission.
Oh,
In
wow.
every situation that we looked at over 100,000 class events, there was really not evidence that just by going to class, you put yourself at risk of getting infected, which was, I think, an important question. It was a useful question to understand how you can balance learning against the background of an ongoing pandemic and be safe.
But it underscores that you do need those protections, right? That the vaccine was protection, the masking was protection. So the idea that you just go out any old where is not proper, but with protection, you are okay.
Yeah, there were limitations to our study, right? We
Sure.
did not have a companion university that didn't require vaccination, that didn't require masking. What I can say with confidence is, at least under the circumstances where we looked at the university, having those in place appeared to be very successful.
Right, right. And that just brings up again, like the measles issue. Remember that a lot of parents gave up the childhood vaccinations around this time, before this time, and there were these outbreaks of measles and so on. And so the vaccination proved to be very important. And may I ask, in terms of in other areas of the country where people were not as well vaccinated as, say, they were in this area, was there a difference, or do you know if there was a difference in terms of contagion?
Yeah, this is a great question. We don't know the answer to it. There were a lot of different approaches. There was a lot of building the plane while we were flying it in
Yeah,
terms of
of
responding to
course.
this pandemic. And I don't think we know the answer to all of those things. The answer that we were able to, the question we were able to ask and answer is a limited one. Right? We know the risk that existed at this one university under certain circumstances, but we know that under those circumstances, it was a very successful situation for being able to do learning, but also to have a minimum of risk inside the classroom.
Right, and presumably elsewhere, as long as people were vaccinated and wore masks, so that really did make a big difference. And then, in terms of your other work there with the engineering of viruses, just give us an idea, if you could, of what you see for future prospects with this. This is an area that many people are not terribly familiar with. It looks fascinating. What do you predict with this in terms of being able to, say, manage disease or improve health generally?
I think from my perspective, the idea of engineering viruses is not particularly new.
No, for
Engineering of
you.
viruses is something that we have done for decades,
Yeah,
and we have done to promote human health.
right.
During the most recent pandemic, during COVID, several of the vaccines that were rolled out were engineered viruses.
Yeah,
They were shown to be very protective.
yes.
There are currently multiple vaccines that are essentially engineered viruses.
Yes,
The FDA approved vaccine to protect people from Ebola virus is an engineered virus.
right.
It is a safe vaccine that is highly protected. And so what I see in the future is that viruses have been and will continue to be beaten into tools
Yeah.
rather than pathogens.
That's very interesting. That's very interesting. And apparently, the big leap was it the mRNA virus that as the structure for these vaccines.
There are there have been multiple leaps that have been incredibly important.
Okay.
In a short format interview, it's very difficult to name all of them.
10 words or less,
But
right?
right. But there are a lot of really incredible advances that have come through the study of viruses that mean that we have more tools to fight disease today effectively
Yeah.
than we did before. And it has come through the understanding of how these viruses work. It comes through an understanding of how they fail and the ability to genetically engineer them in order to make them tools rather than pathogens.
That seems to me extremely important because there is a lot of concern about the spread and development of new viruses, or viruses are going to be that were more localized, like the M virus, the Mpox, will now be worldwide, because we travel and so on. But what you're saying is, while this might be the case, that there'll be more viruses out there, you also are learning so much more about how to control them, so that this engineering aspect is developing in a way that will protect us very much in the future. Is that the case?
Yeah, so that's the way I see it.
Yes.
That when we have researched viruses and we have understood them better,
Yeah.
we have been able to develop ways to make suffering from those diseases less.
Yes. Yeah.
And I see that if there is continuing work done to understand viruses, we will continue to have more and more tools. So it is something where I see immense value in looking at these viruses, understanding how they work. I don't think that if we stop looking at them, viruses will stop coming out of the woodwork.
Yeah.
We continue to be challenged by new
Right.
viruses showing up.
Right.
Or by old viruses spreading to places where they weren't before.
Yeah,
And
right.
so if we just stop paying attention, I think the viruses will count that as a win.
Right. Yes, but you're really onto it now. You have the technology, you have the expertise, and you're really learning a lot, which is wonderful for advanced medicine in our day. Dr. Connor, one last thing before we close. We're almost out of time, but you are at the very famous Needle Lab in Boston, and that is the high security lab. Can you give us a little idea about these high security labs?
Yeah, the basic way to say it is this is a building that it's really hard to get into.
Yes, even for a virus, right?
Yeah, right. And if you're a virus, it's impossible to
To
get out.
get out.
The reason that this building exists is that in order to have those therapies that I mentioned earlier, we need to be able to actually test them in a game time environment,
Right.
and not just a theoretical environment. And so this is a building that was built to do that and has done so. The goal of this building is to find new cures, develop new cures, and make certain that they go all the way from a thought to FDA licensure so that they can be used worldwide. And that's something that we work on every day. It's something that we work on under an extreme level of observation and security and safety and engineering that make it so that we do this work safely, right? This is work that, I commute in here, I go back home.
That's right.
I go back home to my family,
Right.
I'm happy to come home and kiss my wife and hug my daughters in that order. I am not concerned
Right.
about the safety. I am very comfortable with the level of safety in the building and the extremely low level of risk.
Right. I wanted to underscore that. I really appreciate your bringing this out because there was this issue about the security of these the virus labs, and I know that that is the Needle Lab is a very famous building for this, and I hope that reassures the public. That is all that we have time for, and I wish we could go on. This has been most interesting, most informative. Dr. Connor, thank you so much for joining us today, and I wish you great luck with your work.
Thank you. This was fun.
Okay, goodbye.